so in this video, we're going to talk about high energy bonds in the cell. So let's get to it. We talked about, in the previous videos how free energy, so how living organisms are capable of capturing free energy and from various chemical reactions in the cell. And so what do they do with that free energy? They can use it. They can capture it really and put it in a molecule that's considered a carrier of all this free energy. And as we all know, the energy carrier molecule in all cells is ATP, adenosine triphosphate. So which looks like this. I'm sure you've seen it many times in your career. That's the adenosine part, and these are the three phosphates. And so that's the sugar moiety on the ATP, and it's on this moiety that the phosphates are found. And the closest you are to the sugar, the letters start with A, and then beta, and then the gamma. So you have the three phosphates. These bonds on here with the red arrows are the high energy bonds. And it will look like this, the molecule at pH 7. Adenine is the nucleotide base. Okay, so this is how it looks like. Now, I should say that there are additional ones, okay, nucleotides in the cell. It doesn't have to be adenine. It's not the adenine that makes it high energy, okay? It can be GTP if it's guanine, for example. And that's the sugar, which is a D-ribose. The whole, the base plus the sugar is adenosine. It's a nucleoside. And then depending on whether you have 1, 2, or 3 phosphates, you have AMP, which is just the alpha phosphate, ADP, alpha and beta, and ATP, which has all 3 phosphates, including the gamma one. So these high energy bonds are really phosphate anhydride bonds. And what does that mean? It's really 2 phosphates bound to the same oxygen. So these are the 2 phosphates bound to the same oxygen. So imagine that this would have been water if those were protons, but they're phosphates here. So high energy, there's nothing really special about these bonds. It does not refer to the bonds themselves, but really to the free energy that is released if the bonds are broken, if they're hydrolyzed. And why there is so much free energy released? Because the products have much lower free energy than the reactants. Okay? And for every one of these phosphate anhydride bonds that you break, that's the delta G that you get, about minus 7.3 kilocalories per mole. Again, delta G, if the sign is negative, that means you have a release of free energy. So it's completely opposite again from the delta E, which had to be positive for being thermodynamically favorable. I should say that, again, the 7.3 kilocalories per mole is at standard conditions, which, as we have seen, they are far from real conditions in the cell. So again, there is nothing unique about the bonds themselves. Why are they high energy bonds? In all of these ATP mediated reactions, there's a magnesium that coordinates the phosphorus, and it makes it more electrophilic. Okay, and all the phosphoryl transfer enzymes, they have magnesium at their active site, so that they can actually facilitate this reaction. So what happens when the bond is hydrolyzed? Okay, why is it high energy? So for several reasons, okay, what happens is that you reduce the number of negative charges that are next to each other. So of course, things of the same charge, they repulse each other. You have all these negative charges on the phosphates, and that's unfavorable. Right? And for example, you see here, okay, they have one negative charge here, and here. So the moment you hydrolyze this bond, okay, so all of a sudden you have reduced the number of negative charges that are close to each other. And this is how, that's one way, not the only way, as we'll see. That's one way that you release free energy. So the 1-phosphate, so whatever gets split here, okay, is, so the water will attack the phosphate, and the hydroxy, so one oxygen and proton from the water will be attached to the phosphate, and you will end up actually with an inorganic phosphate. and the other proton from the water will be picked up by let's say, by the beta group here, and it's going to look like this. They are called phosphoryl transfer and not phosphate transfer because it's really one phosphate and three oxygens. This part here, the highlight, that gets transferred. Okay? If it's a hydrolysis, you end up with a phosphate, as I said in the end. All right, that's one reason. So that you have a release of free energy. Again, you minimize the electrostatic, or you reduce the electrostatic repulsion upon hydrolysis. The second reason is that you also allow the electrons of the phosphate groups to delocalize more. So what do we mean by that? So take, for example, the phosphate that's generated here after hydrolysis. You have the phosphoryl and the hydroxy group from water makes you a phosphate. And the more you can bounce around the electrons, okay, on the phosphate or on any molecule really, the more stable that molecule will be. And there are many coordination states, and so you can move them around. But there are many ways that this can bounce. For example, in the phosphate, and in the end, you delocalize all these electrons in a nice way, and you did not have these options when the phosphate was bound to ATP. When it got released, now all of a sudden you can delocalize better, and that makes it more favorable thermodynamically. Lastly, the ADP that is produced immediately ionizes and releases a proton, and that's favorable in a solution with few protons. And so the proton that got picked up, as I said, by the water, immediately you're going to lose it. So you're going to end up with your ADP will look like this. So you have a release of a proton, which came from the water, and that's a good thing at pH 7 because you have almost no free protons there. And the fourth reason that this is a high energy bond, it releases a lot of free energy when it gets hydrolyzed, is that you allow more water molecules to bind to the products, phosphate and ADP, compared to the number of water molecules that could bind to the reactant, which was ATP. Okay? So for all these reasons, all these four reasons, this is why ATP hydrolysis releases so much free energy. So we mentioned that under standard conditions, the delta G for ATP hydrolysis is minus 7.3 kilocalories per mole. But that assumes again that everything is present at 1 molar, which just is not going to happen. In fact, the real concentrations, let's say in red blood cells, is more like this. So you have 10 times, as it turns out, more ATP than ADP, and we'll talk about this later, why that is. And at millimolar concentrations, not molar. So for all of them, actually, you have 2 point something for ATP, 0.25 millimolars for ADP, and the phosphate concentration is close to 2 millimolar, also 1.65. So if you know this now, you can go to our equation. Okay, delta G equals minus RT ln and the natural log of the equilibrium constant. And the K here, you can calculate that for the actual amounts you have, not 1 molar, everything, right? And if you do that, the actual delta G at non-standard conditions, where you have these concentrations, is almost double. Okay, it's minus 12 kilocalories per mole. So you release 12 kilocalories for every mole of ATP you hydrolyze. So you would think that's great. You get so much out of it, so much more out of it compared to the minus 7.3 kilocalories per mole value that you had for the delta G under standard conditions. But of course, to make it, you also require a lot more energy, right, under these conditions. So in the end, it evens out. So the key point that I want to make is that the ATP is not used for storage of energy. Yes, it has these high energy bonds, and it can carry them. It carries energy in its high energy bonds. But the better way to view ATP is money. It's like money. It's a currency, the energy currency that all cells use to do their energy transactions in exactly almost the same way that we use money to do all our transactions for goods and services, let's say. So Why is that good? You know, just like money, you know, you pick up $100 bill from somebody else that gave you, all of a sudden now you have added to your free energy the ability to do useful stuff, useful work with your $100 bills. Likewise, you move a phosphoryl group, okay, from ATP to another compound, and that compound now has more free energy available to it if it gets involved into some chemical transformation, okay, for some other reaction. So this phosphoryl transfer, we saw hydrolysis only, but actually the phosphoryl group can go on to other compounds, as we will see many times in this class. And that makes these phosphorylated compounds very useful to the cell. It makes them very useful for various other chemical reactions. Again, just like money. So at equilibrium, would you expect, you have a high energy compound, ATP. This is a key point here. You have a high energy compound, ATP has a lot of free energy. Would you expect to have at equilibrium more ATP or ADP? The obvious answer is that you would expect to have a lot more ADP compared to ATP, right? In fact, at equilibrium, the ratio is astronomically low, 4 times 10 to the minus 8. But cells, as I just showed you for the example with red blood cells, so here again, okay, they have about 10 times more ATP than ADP. Okay. So what does that say to you? So it says to you that cells are far, far, far from equilibrium. Okay, it's a huge displacement, almost a billion fold displacement from equilibrium. And why do they do that? That enables them to drive reactions that are unfavorable. Okay? So they can actually make things happen, the cells can, by keeping the ATP to ADP ratio so far, far from equilibrium. Okay, and they go into a lot of trouble to make an ATP and to maintain this very thermodynamically unfavorable ratio, so they're far from equilibrium. They are in steady state again. Remember, living systems are all in steady state. They maintain this ratio very close, actually. Here, this one, ATP to ADP, within this very relatively narrow range, but which is far, far from equilibrium. So that goes back to the previous point that we had made in the previous video, how all living systems are in steady state most of the time, but that doesn't mean they're in equilibrium. They're far, far from equilibrium. So how do they make ATP? All right, so it's useful. You know, what's the money tree that they can go pick up, money, currency, and do their business? So cells can make ATP in 3 ways. The first, probably one of the most ancient one, is or probably the most ancient one, is a substrate level phosphorylation. You simply have simple chemical reactions and you move around phosphoryl groups, let's say. Okay, and this is what happens with glycolysis. It's a super ancient way to make some ATP. The way to make a lot more ATP, okay, is through electron transport phosphorylation. So this is oxidative phosphorylation, and what happens in this case goes back to the redox reactions here, is that they move electrons, which they capture from food, okay, and they move them around across a membrane. Now, because they move this electron, they couple the movement of these electrons from some terminal acceptor, in this case oxygen, let's say, in most cases like our cells. This movement of electrons is coupled to some gradient formation across a membrane, okay? Usually by protons, but it could be other ions too. And in a sense, they couple movement of electrons to a terminal acceptor to the charging of a battery. Okay, so you have a voltage difference actually between the 2 sides of the membrane, the outside from the inside. And when you have a charged battery, as we saw in the previous video, you can do work. You can couple that to actual work, and that's how they make most of their ATP. And the machine they use to make ATP relies on the movement of these ions across membranes. Okay, so we'll cover this part. So the substrate level phosphorylation glycolysis will finish before exam 1, and between exam 1 and exam 2, so we will be talking about electron transport phosphorylation exclusively. The third way, however, actually not explicitly, I said, there is another way to make ATP, and that's for the phototrophs, that's photophosphorylation. And it's similar to the electron transport phosphorylation in the sense that, again, electrons play a key role. And how you get high energy electrons in this case is through light. You use light to raise the energy of these electrons. But again, the mechanisms are very similar with oxidative phosphorylations, because the movement of these electrons then along an electron transport chain leads to the generation of ions across a membrane, protons also in this case. So again, you charge the battery. So you use light to increase the energy of some electrons, and as the electrons move to some terminal acceptor, now you charge your battery in a sense again, and you can use that voltage that you generated to drive formation of ATP in exactly the same way as in ATP synthesis by oxidative phosphorylation. So these are the three ways that cells can make ATP. I already mentioned this earlier, just to keep the names correct. If you have three phosphates, you have a phosphoryl group. Okay, this is actually what immediately you get when you transfer the gamma phosphoryl group of ATP to something else. If it's water, it will be converted to a phosphate. Okay, but it doesn't have to be water. If you here you have two protons, that's water, but it doesn't have to be. It can be X, anything. Okay, and that X now, okay, the oxygen, okay, of that X will be phosphorylated. Okay, so the X itself will be phosphorylated and will generate ATP. X could be a protein, could be a lipid, could be a sugar, could be, you name it, almost every biological entity, okay? Every biological molecule. If you move both the alpha and the beta, then you have pyrophosphorylation. Okay? You don't have to move just the gamma. You can move both of them, the gamma and the beta. Okay? And of course, what's left from the ATP is AMP, just the alpha phosphate. There's something else that we're going to see repeatedly. And it doesn't happen only with ATP, but it happens all with the other nucleotides. It could be UTP, it could be GTP, for example. That X, which now doesn't have to be oxygen, but it could be a nitrogen too, if it attacks the alpha phosphate, okay, then that X gets to be, if it's ATP here, adenylated. So the whole part here, okay, the nucleoside plus the alpha phosphate will be covalently attached to molecule X. Okay. And the two terminal phosphates, the beta and the gamma, will be pyrophosphate. Okay. This is an extremely favorable reaction, because the pyrophosphate, okay, so you see the delta G here is minus 10 kilocals per mole, more than ATP hydrolysis, because the delta G will for the first reaction, because, and then even more than that, the pyrophosphate will be immediately, it's very unstable, so it will be hydrolyzed to give you two phosphates. You have a huge release of free energy. This is done to drive very unfavorable reaction. Really, you are paying a lot, okay, to activate this X. You decorate this X with the AMP essentially, okay, and in that way you have endowed, it's like you gave it a lot of money. So now X, thermodynamical spin was very poor, couldn't be involved in too many reactions, but once you adenylate it, now all of a sudden it becomes useful. It can do chemical reactions. So what compounds are those by ATP? There are many, as I said. They can be phosphorylated, they can be pyrophosphorylated, they can be adenylated. And once any one of those in this order, okay, so from lower to higher, gets activated. So adenylated is much more activated than simply phosphorylated. And you can use these modified now compounds in various metabolic reactions. So ATP is made using energy, as I said. If it's, let's say, food, let's say glycolysis or oxidative phosphorylation, in organisms, or by light. If you use light in prototrophs, that's how you make it. So you use the energy either of food that you consume or light, if you are able to capture the light. And again, you hydrolyze it now in order to drive thermodynamically unfavorable reactions. And what are those thermodynamically unfavorable reactions? Transport and motility are very simple. I mean, you can understand, to move, you have to spend some energy, right? Biosynthesis is also extremely unfavorable. In biosynthesis, you make things that are bigger from smaller building blocks, and every time you build something, you have to expend and to spend energy. Okay, you have to spend your energy currency, which is ATP hydrolysis, to drive that. Otherwise, it wouldn't happen. It's thermodynamically very unfavorable to make big things from smaller ones. On the other hand, when you consume and digest your food, you break down big things to smaller ones, and that's favorable. And you can, if you're able to capture some of that free energy, then you can make ATP. So that's a general scheme, and you use from the food you eat, or if you're a photosynthetic organism, you can break it down, and you make ATP in the end. And ATP is like money, and you have to spend it. Okay, it doesn't do you any good to keep it around in huge amounts. And how do you spend it? Mechanical work, biosynthetic work, transport work. Here I mean moving things in and out of the cell. Okay, this again, it's an open thermodynamic system, and things have to come across the cell membrane in both directions, and that usually takes some ATP. So I already said that you make ATP and you have to spend. Doesn't do you any good to keep it around. Again, just like a bank. Okay, a bank is not a safe where you can put all your money and be there forever without anybody touching them. In fact, the bank will go out of business if it's like this. What the bank does and grows is to lend the money. It keeps some, but there is a turnover. Okay, so you can lend it and then it gets more, and so on and so forth. In fact, if at any given point you ask how much ATP do I have in my body, you would have maybe 1 or 2 pounds at most, usually around a pound. Okay, but how much do you actually turn over or use every day, make and spend every day? On an average day, you turn over about your weight in ATP. Okay, so it's a huge amount of ATP transactions. And even though at any given moment you only have a small fraction of that as ATP. All right, so ATP is very useful. It's the major one. There is some other phosphocurrencies, again, phosphate-related, phosphorylated compounds that they have similar roles. By the way, hold on a second. Yes, so we'll talk about this in a minute, but if it's so good, why can't we just, well, I mean, we can synthesize ATP, right? I mean, the ATP synthesis has been done since, for decades now, since the 50s. I believe the first person who did it was Kalkar. So why can't we just eat ATP and satisfy all our energy needs? Well, it won't do you any good. And, ATP, we have some bottles, some vials upstairs with ATP, is extremely negatively charged. So you eat it, okay, it will never go through your membrane. Okay, so you will never be able to absorb it, and it will just be excreted out of your digestive tract, and you got nothing, right? Anyway, what are the other high energy metabolites? So we've seen ATP, the delta G, ranked here in terms of the delta G when the phosphoryl group is hydrolyzed. So ATP is right in the middle. Phosphenol pyruvate is double the delta G. So a very unstable compound, as we'll see. Once you hydrolyze that, you release a lot of free energy. Okay, so that can be used to drive a lot of things. 1,3-bisphosphoglycerate is also pretty high up. Both of these things, we're going to see them in glycolysis as intermediates in the phosphoryl transfer reactions. I already told you that many compounds get modified by phosphorylation. This is glucose or glycerol, for example. and they can be phosphorylated at various positions, either the first carbon or the sixth carbon. And again, if you hydrolyze that, you do release some ATP, some free energy, okay, but not as high as ATP itself. The point here is that again, the phosphoryl group endows um, um, the, whatever is phosphorylated with it makes it richer energetically speaking, right? It's just like money, so it endows it with free energy that can be spent for other reactions. So ATP seems to be the chosen one because it's kind of right in the middle here. It's very, very conveniently placed to, you can make it easier and you can spend it. It's not so easy to make phosphoryl pyruvate, for example. It's very energetically unfavorable. So maybe that's why it's the chosen one. Why phosphometabolites? They stay inside the cell. Why phospho in general, right? As I already alluded to, they are negatively charged, so they cannot get out of the cell. They can also usually don't get in by themselves, right? So once you stick that negative charge, it's yours to keep, right? Now, and every time you do the phosphoryl transfer, the actual phosphoanhydride bond is maintained, so you don't lose it. So you can, again, it's just like currency; it retains its value, let's say. In addition, once you phosphorylate something, whether it's let's say simple sugar like here or another compound, or a protein or a lipid or something like this, which can also be phosphorylated, immediately it's like you plant a flag on it and it can be seen as phosphorylated. In fact, there are specific enzymes that only bind the phosphorylated versions of their substrates and not the other ones, right? So it has many useful properties, and it's fair to say life wouldn't exist, or if it existed, it would be very different compared to what we know about it if it wasn't for phosphorylations. But it is not the only high energy compound of all these phospho compounds. In fact, a super ancient one is a acetyl-coenzyme A, which we're going to see a lot also in this class. So the coenzyme part, so this is, it's a long and complicated molecule. You can see some similar, let's say, modules like we saw in ATP. There is the ribose, there is the adenine. So reflecting some ancient origins of all these things. But this one has a few more. Okay. So the acetyl group is right here at the very end. This sulfur is the business end of the molecule. The acetyl group is attached here. And it's really the key compound that is used to move around carbon units. Okay. Acetyl groups. Why is that? Okay, so if you have acetylated coenzyme, so coenzyme A would be up to here. If it is acetylated, it would be acetyl coenzyme A. If you hydrolyze this bond now, so this is an ester, but it's not an oxygen ester. It's a thioester. Okay? So if you hydrolyze it, you get coenzyme A. Okay, there it is. So that's the hydrolysis. You have coenzyme A, and you release acetic acid, which then loses a proton and becomes acetate. So you can delocalize here the negative charge on the two oxygens. But the product, okay, has the same energy as an oxygen ester. Why have this sulfhydryl here? We can have any other regular, vanilla oxygen ester, and the product we get is exactly the same. It's acetate, right? So why does hydrolysis? But hydrolysis of an oxygen ester does not release so much energy as a hydrolysis of a thioester. So why is that? Because even in the unhydrolyzed state, the charge here can be still delocalized. Okay, so the acetate itself, it doesn't gain too much when it's by itself. Okay, as opposed to the oxygen ester. But this option does not exist for the when it's in the thioester configuration in acetyl-CoA. So the negative charge here cannot delocalize. So it's more unstable. It has a high energy, so it wants to be hydrolyzed. Okay, this one couldn't care less because again, it's the ester is perfectly stable, the negative charge can be delocalized, and it makes no difference. So we talked about now the energy currency. I think we will stop at this point with this video, because then we have to talk about another currency, which is the reducing currency. We talked about electrons, a couple of videos ago. And so how do the electrons get carried around, the reducing power? So for that, we will continue in the next video.